Today is basically the DNA lecture. We'll have a lab on DNA today. And, well, I call it cytoplasm and protein synthesis because that's kind of what you have to know about for cells that make proteins, and that's what DNA does. It carries instructions on how to make proteins. So I kind of start off lecturing on the different cytoplasm components, then we kind of get into DNA and protein synthesis. So the basic idea of the cell is cytoplasm still on chapter 3, and I gave you a fair warning about homework 1. But guess what's coming Wednesday? Lecture exam 1, which also covers this material, which I'm sure we're clear. First exam covers chapters 1, 2, and 3, what we talked about. Today's lecture, cytoplasm, pretty much from here to here. All the cell organelles which will be contained between the cell membrane and the nuclear membrane. All cytosol and cell organelles contained within cell membrane, but outside of the nucleus. We'll focus mostly on the cell organelles. The cytosol is just gelatinous stuff that things are embedded in. It's basically the ICF. And the mitochondria is important for the generation of ATP. And I already mentioned this about it. Here's a nice picture of it. That'll be the first cell organelle I cover. In high school textbooks, we always call it the powerhouse of the cell because, it generates the ATP. And I'm not going to get too much into the cell metabolism that I mentioned before. I don't know if you remember when I gave my intro lecture on cells, the events that occur in the mitochondria, it's not glycolysis, it's the aerobic metabolism stuff, which would include, go back and look at your notes, TCA, tricarboxylic acid cycle. It accomplishes that, as well as the electron transport chain. And I said that these were aerobic metabolism. Okay. That's all I'll say about it. That's the cell metabolism that gives you a lot of ATP. Structure-wise, it has a double membrane because all these chemical reactions require lots of enzymes. And so this inner matrix called the cristae, let's see, I'll just say it's studded with enzymes. For TCA and electron transport. I don't think I mentioned that double membrane before. And cells that have lots of mitochondria, like stomach, they must be doing a lot of pumping or transporting or something, because that's why they need them to do work. There's a picture of a stomach cell which is pumping protons. And also cells that execute a lot of protein synthesis are going to need a lot of ATP. And that's really what I'll focus on today. The idea that cells synthesize proteins. Okay, that's crucial for a cell biology chapter. So I spend the most time talking about this. So proteins are not only a major structural component of cells, but in the form of enzymes that mediate every process in the cell. And understanding the steps of how proteins are made is important. And what organelles are important in protein synthesis. So you need the mitochondria for the ATP, but they don't directly participate. So I kind of outline the process here. First thing, you have to code for the protein, which is in the nucleus, which contains the genetic code, the DNA. You need that correct amino acid sequence, and it's contained in the genes. That's the first thing. Where is the code for the protein? Whatever protein you're trying to make. Maybe it's the sodium potassium ATPase bond. I don't know. It could be anything. But the cell is supposed to make it. Maybe it's supposed to make hemoglobin. Well, the code is the amino acid sequence. Just the sequence. Now, test question. What order of protein structure is that? Just the sequence. Primary. Yeah. So, that's the code. And the primary structure, put that parentheses. So that amino acid sequence, the code is in the DNA. But what is DNA? Is DNA amino acids? No, DNA is DNA. DNA is nucleic acids. So for DNA, the code it's in the the nucleic acid sequence. We'll kind of get into that. I haven't taught you the structure of DNA yet, but the code's in there, and it's the job of the cell. RNA really does it. The different kinds of RNA can help synthesize the polypeptide chain, your protein. Remember what a polypeptide chain is? It's a bunch of amino acids linked together. Okay. So you have your 3 RNAs. Help this process. Now, I don't have them listed in any particular order, but that middle one, let me talk about that one first. That's the messenger. Okay? mRNA. mRNA RNA is like DNA. Okay, structurally they're very similar, and so they can interact with the DNA. This molecule can interact with that molecule, and when you generate this, it's like it's going to have that code. It's going to take that message so you can kind of translate it into the correct amino acid sequence. And so it's very important to have a messenger, because the code is in the DNA is in the nucleus, but you can't make the protein in the nucleus, is the thing. So that's why you need a messenger to get it out of there. So if you have DNA, which is a double coil thing, one of the things you have to do, like I drew here, you got to open the gene. You have to expose this double helix thing, so you can expose one of the nucleotide sequences, so you can read that code. Okay, so pretend you're this is all within a nucleus, and this is all within a cell. I got to make this protein. Here are the instructions right here. So what an mRNA does is like, it'll, very simply, I'll use a different color, but it'll just kind of like, you know, read it, link up all these nucleotides, and it'll just kind of keep reading it. And by getting the correct nucleotide sequence, it's got the code. This molecule, the mRNA, which I drew as a red squiggly line, and then be a messenger and take it into the cytoplasm. You got to read that code. And our RNA can kind of combine with it. We call this the, you know, kind of the cytoprotein synthesis. Now we're in the cytoplasm. I'll just put pro-synthesis. You know, our RNA has kind of like a small and a large subunit. Okay, and it'll just kind of read along with that nucleic acid thing. The problem is, we got to go to proteins, but we're in nucleotide form now. Okay, and I'll teach you what the genetic code is, but we got to decode it. Okay, like if you read, I can only read English, but someone gives me a book in Spanish, I need to translate it in the language I can use. Cells don't use nucleic acids, they use proteins. So you have to translate it in a usable form, in a usable molecule that the cell can use, proteins. So you had to be translated from these nucleic acids to amino acids. Now, transfer RNA can help with that, because transfer RNA is an RNA that can kind of match up with the mRNA, and it'll transfer the correct amino acid for the code. Okay, so it's like you get your tRNA. I'll just say the T stands for transfer, right? I'll just say transfers the correct amino acid. So maybe there's a, you know, tRNA, and it's carrying an amino acid to it, and you kind of like take it here, you know, and a whole bunch of them are going to come, and I don't want to try to draw the whole thing. But the point is, you've gone from just kind of transcribing the gene to translating the gene out here. That's the correct term. So when you read the code, you get your mRNA transcript. It's just the correct sequence of nucleotides. That's transcribing the gene. Transcription. But you didn't make anything yet. You made an mRNA, but that's not making the protein. But that's the code on how to make it. Remember what the code is, the correct amino acid sequence. So when you get over here, now you're decoding it and you're translating it into amino acids. So this is called translation out here. When all three mRNAs kind of come together. Okay. So once you're done making the polypeptide chain, you have to process that protein so it can be secreted or inserted somewhere. And then so we kind of talk about, oh, less so, the endomembrane system. Okay, there's too much to draw here, but it's ER and Golgi. I'll get to that. Here's kind of a professional picture of what I was trying to draw. They leave out some details. They trim up the mRNA. But they do show you the basic idea of transcription and then translation. So you got to go from the RNA, from basically DNA to RNA to protein. That's the basic idea that you need to take away from this lecture. There's the polypeptide chain there. So let's look at a professional picture of the nucleus instead of just a circle on the board. You should get some detail as to what it might look like under a real scope. Now, it contains the DNA. That's the most important detail we should know about it. And what we learned in mitosis is during cell division, you have to package the DNA in a way so it doesn't get damaged, and put it during mitosis. DNA kind of supercoils with histone proteins. And you can kind of view these in the form of, the chromosomes. The DNA, the genes are in there. So whenever you hear the term genes, it's a discrete segment of DNA within your supercoiled DNA. You got 23 chromosomes, 23 pairs. You inherited half a set from your mom, half from your dad. Genes is a term that came well before Watson and Crick. I think Gregor Mendel, with the pea pod plants, he coined the term genes hundreds of years ago. And we always study that in terms of inheritance, but it had nothing to do with DNA. They didn't even know about it. And the concept of the gene is you pass some trait on to the next generation. You do the Punnett squares and all that stuff. We don't do that in this class. But that's where the term gene came from. We still use it, even though we know now it's in the DNA. The other thing I guess I should note is how this nucleus is porous. That's important because, messengers can get in, mRNA can get out, things have to enter and exit. Hormones can enter. You want to turn genes on, things have to bind to the DNA. mRNA exits. So things can enter and exit. You can see a nice picture of the pore there. Here's a picture of the chromosome. And I like this figure. If you kind of start at the top, that's kind of the microscopic view. It's all microscopic. But you can see how it intertwines with the histones, and they're kind of all wound up in the visible chromatid there. What we can view under the microscope. Well, we did that in our first microscope. Now, I don't know if you noticed that they kind of drew an ER with the nucleus. Because a lot of times the protein synthesis will occur right outside the nucleus, right here. And that's actually the ER. And the endomembrane system includes the ER and the Golgi. So this is just the ER part. Endomembrane system. includes the ER, which stands for endoplasmic reticulum, a reticulum is a network, and that's what that means. Endo means within, and there's rough and smooth. Rough ER has the ribosomes. Ribosomes is another alternative name for the RNA. And then smooth ER doesn't have the ribosomes. I'll say it without ribosomes. The function of the endomembrane system is not pictured here, but they will receive newly formed polypeptides, okay? And it'll help process them for excretion or exocytosis or whatever. That's the function. I'm gonna go over here. Receive newly formed polypeptide chains. Okay, and when it does so, that's where the protein will fold. It'll take shape. You can glycosylate it in there. You can kind of get it ready for its protein function. Proteins fold. I said they're glycosylated. You're basically processing it, and also you're going to hand it off for more packaging and exocytosis, the Golgi, shown here. So the Golgi apparatus is the green structure shown here. It's receiving protein vesicles from the rough ER, which pinch off, and then those membranes fuse with Golgi, and those proteins are then modified within the Golgi compartments. What modifications? I'm not really concerned with it. They are modified a little more, so you can package it. Either they're secreted, or maybe you're just making more cell membrane, or maybe those proteins are for lysosomes. Okay, that's what it's shown there. So, let's say you have ER, this cavernous thing. Your little protein vesicle that got pinched off, you're being handed off to Golgi next. Looks like a stack of pancakes here. And you're going to fuse with that structure. I'm writing cis and trans. Biologists refer to the side receiving there as cis, side opposite trans. Or sometimes they call it, instead of cis, they'll call it forming phase, depending on what book you read. Or maturing phase on the other side. So that proteins will be secreted. These proteins will be handed off in this direction, and they'll be secreted by the cell. So what I drew in red was ER, and what's green is Golgi. Okay. So what's pictured there, what I drew here, when I say endomembrane system, it's this whole thing. Now I want to move on from that and just talk about the lysosomes themselves. I alluded to them in the previous slide. Their function is outlined here. Now they're filled with digestive chemicals. A lot of times, a lysosome, the cell will take in some kind of debris. No, I'm sorry, no. This function here, number one, you merge it with the damaged organelle, maybe that's a mitochondria, and you get rid of it. Destroy damaged organelles. That's one function. Another function is to, this one here, we would take in some piece of garbage, and then a lysosome is merged with that vesicle for breaking it down and getting rid of it. Destroy phagocytized materials. The third option shown there is that the lysosomes kind of burst and all of their lytic enzymes will actually destroy the cell. Now that's called apoptosis. That's a programmed cell death, which is, you're supposed to do that. Apoptosis, programmed cell death. So lysosomes can participate in that function. And the other thing about cytoplasm, the last things I think I talk about here, is the cytoskeleton. Okay. You have all different kinds of microfilaments, and I don't spend too much time talking about them in this chapter because when I get to tissues and connective tissues, we talk about the fibers a little more there, and we even see them under the microscope. But at least the pictures give you an idea of what the cytoskeleton is. The one I want to mention right now is microtubules, because I've been talking about them a lot, at least in mitosis. Microtubules. But microtubules do more than just form the cytoskeleton. They, I think in this class, we kind of see them as additives to cells in the form of cilium too. So microtubules form cilium. That's singular. Cilia is the pluralized form of the word. You usually see this on epithelial cells. That's kind of where we'll see it most. you have a generic cell, at the apical surface, these microtubule structures will kind of like be stuck at the top. And they have an organized whipping that they can do. If you look at a cross section of it, that's what this slide shows. There's like a 9.2 and a triplet pattern form. I don't care about the pattern. Just know that they're cilia. They're made of microtubules, and we'll see them on epithelial cells. I did talk about centrioles in terms of the centrosome matrix, so they form that as well. So I'll continue over here. And the last thing that's worth mentioning are how microtubules can form a system where the vesicles can work with motor proteins to move the vesicle in an organized way throughout the cell. So microtubules work with motor proteins to move vesicles. That's why I put this slide in to remind me to mention that. Okay, let's move on here. There's the picture of the whipping action of the cilia. This is most important in the trachea. The lining of the trachea has this columnar epithelium where you can beat mucus in one direction so you have a productive cough. For example, a mucus layer on top of an epithelium will look like this. And the arrow is pointing in one direction because a layer of mucus which catches the debris, and with coughing you can kind of get the stuff out of your upper respiratory tract. The last thing that's not made out of microtubule, but it's a structure that we can see on cells, is microvilli. So I'll draw a big line. Microvilli aren't microtubules at all. They're a superfolding of the plasma membrane. Microvilli. Folding of the cell membrane. You're only going to fold the side that's active. So microvilli isn't something you stick on the top. It's actually the cell membrane folded. So if this is a cell with no folding at the top, a cell with folding at the top would, be like that. That's microvilli. Microvilli. And what you're doing is you're increasing the surface area for function. So on that side of the cell, maybe things can be absorbed and secreted through that side, and since there's more surface to work with, well, it just makes it more efficient. So if these things want to come in, well, I guess the word is that functions for absorption and secretion when you increase the surface area. Better absorption and secretion. Okay, let's move on. So I was trying to get to DNA a lot. I finally got to it here. So we're going to move away from cytoplasm and talk about DNA. And for this part of the lecture, what I did was instead of like using pictures from the book and quotes from the book, I kind of follow their original paper that they came out with in 1953. So I think Watson and Crick are still household names, I think. I think even students have heard of them before. The duo that, they didn't discover it, they kind of elucidated its structure. There have been a lot of people who've been trying to work on it, Linus Pauling and everything. Well, anyways, there's their model. See their model they're standing around? You're going to build that today in class. Well, I mean, not one so fancy, but yeah, it's like, if you can do it, I think you'll kind of learn about what the structure of DNA is. So basically, you got an old guy and a young guy. Which one's Watson? The old guy or the young guy? People always say the old guy. He's the young guy. Now, who do you think was more brilliant? The old guy or the young guy? The young guy. He was 23 when he did this work. He was super, super off the chart, this guy. So he's still around today. I mean, he did a lot of contributions in proteins and other things, and he's probably the biggest name in biology, and he's still around. And, I can remember all my profs talked about him when I was in grad school. And he was key in this whole thing, where DNA is the primary genetic material, and that was the big news, 70 years ago. And the idea was that genes determine the structure of proteins, because that's what geneticists were wondering, right? If you're a geneticist and you don't know if even molecules could if genes are in molecules at all, they didn't even know that. And they did, and then they figured out it was DNA. As recently as 1950, the molecular structure of the gene was unknown, so it was impossible to explain the gene-protein relationship. In the classes of molecules they wondered about, they actually thought it was proteins, that proteins had to have the genes, because they were much more diverse and numerous. And they knew about nucleic acids, they didn't think it was that. But in big discoveries, what most people believe are usually wrong. And so that paper that came out in 1953, I have the first page there. Now you think for something so big, it'd be a huge paper, and it wasn't. That's it. That's the paper, one page, and there's a couple of paragraphs on the next page which I did not include. So you could sit there and read it and understand most of it in about 5 minutes. Okay, and it's probably the most important paper that came out in the past 150 years. But anyways, I put quotes from it that I think we could understand here today. Because one thing about Watson I noticed, some scientists when they write, you can't even understand what they're saying. I've sat through meetings where like the senior researchers, like I read that paper, I don't even understand it. Because like scientists are so erudite, when they write about their own work, it's like they get on their high horse and you can't understand what they're trying to tell you. So what's the point of writing? Watson was not like that. He actually made his writing very clear so even the layman can understand his work. I mean, if you want people to understand your discovery, you should write about it so people can understand it. And he was very good at that. Like, here are some of the things he said. DNA has a highly regular shape. I think most people can understand that concept. It's right-handed. Like, you'll kind of get that sense, how it twists to the right when you make your DNA model. And hydrogen bonds. You got hydrogen bonds between base pairs. You learn those terms in basic chem. They have a little picture in the inset there. But he's a very good writer. And I like how we went through the terms like transfer RNA, rRNA, messenger RNA. He uses words like transfer and messenger to describe the molecule's function. He doesn't give them some weird fancy name that we had to memorize. It means nothing. So he made it very usable for us. So here's a picture from your textbook. Okay. The quote is from the paper. It says, DNA is a coiled double polymer of nucleotides with an alternating sugar phosphate backbone. So that's his description of this molecule of the model that he made. Okay, let's break it down. So it's double, it's not single, so we got that. Polymer means it's got a bunch of parts linked together. Okay, polymer, not a monomer. It's a double polymer, not a single, it's a double. So you're going to have two strands, right? Because it's a double. Yeah, it's a double-stranded nucleotide sequence composed of nucleotides. So those are the building blocks of DNA. That's what it's a polymer of. But in addition to the nucleotides, which we spend the most time talking about, the nucleotides are like, see, like if you uncoil that, it's like a ladder. The nucleotides are like the rungs of the ladder. And it says that these rungs of the ladder, these nucleotides, are kind of held together with hydrogen bonds. But there's also alternating a sugar phosphate backbone, kind of the, right there, it says right there, these deoxyribose sugar phosphate. Sugar phosphate backbone. Well, there's 4 different nucleotides. Well, actually 5 if you include the one for RNA. Well, anyways, what it says here is the basic structure of a nucleotide. There's a nitrogenous base with a phosphate and a sugar group. Let me write it on the board there. That font's kind of small. Oh, nucleotide structure. A nucleotide will have the sugar, whether it be, ribose or deoxyribose. The phosphate. Nitrogenous containing base. Now the bases could be one category of purines or pyrimidines of base. So it could either be a purine or a pyrimidine base. And the purine bases include, you don't have to know the structures, but remember the names, adenine and guanine. I'm going to abbreviate them as they're commonly abbreviated, A and G. All the other ones were cytosine C, thymine T. Those are found in DNA, and you'll have uracil and RNA only. So cytosine, and then thymine in DNA, and then uracil in RNA. So you got these bases, and another major concept is base pairing. You have specific base pairing via hydrogen bonds, the rungs of the ladder, right? So this is a direct quote from the Watson and Crick paper. If adenine forms one member of a pair on either chain, well then the other member must be thymine. So A will always bond pair with T. And then similarly for guanine and cytosine. So C and G always kind of bond together, these hydrogen bonds. A with T, and C and G always together. If only specific pairs of bases can be formed, it follows that if the sequence of bases on one chain is given, then the other, then the sequence on the other chain is automatically determined because of this pairing. So I just kind of made up some letters and put them in a row, and you should be able to figure it out if I give you one sequence, right? So what do I have up there? TCG CAT. If I give you one nucleotide sequence, the other is determined, right? Because T always goes with A, G, C, G, T, A. Oh, I determined the other one. So if you know how the bases are paired. So that becomes important when we have conversations about whether it be, say, replication or transcription, because in either case, you got a base pair. Okay, so let's say this is I'm talking about replication. You need to replicate the DNA in the S phase before you begin mitosis. I think I taught that. So if the goal is replication, let's say this is the old DNA. You gotta replicate this old DNA. I don't know if old is the correct term, but you want to make two new ones. So you got to replicate this thing so you get from the old you get two new. So what you do is you kind of separate all these hydrogen bonds here, and you kind of go like, so T C G C A. Just kind of rip them apart. Now you've exposed the sequence, and now you can replicate. So you just kind of base pair this one. T, C, G, C, A, T. Right? And do the same thing with this one. Replicate that. I don't think anyone's having a problem so far. It's pretty basic stuff. Now, do you see how we've made two new DNAs? But do you see how we've kind of held on to half of the old strand from the previous one? We've conserved it. We didn't get rid of it. We just kind of used it as a template to make the new one. So your new DNA, I guess new DNAs, yeah, two of them, right? We've conserved part of the old strand. And so we say this process is semi-conservative. DNA replication, tag it on here, is semi-conservative. But it's the whole concept where if one strand is given, the other one's automatically determined because of the base pairing. Here's a picture of DNA replication, and this cannot happen without enzymes. So what you see here, I think what I'll note on the board are some of the enzymes you should know that help this process. Well, you unzip it this way. There's a kind of like a 5' 3' thing, and I'm not really concerned about that. It's not even on the figure. But to unwind it so you can kind of expose your templates there, that is called helicase. So that's one enzyme that's important. To actually make the DNA for both strands, it's a DNA polymerase. You can only make it in one direction. Okay. So notice that the replication fork, where it's unwinding towards the left, right? Correct? And so this one here, the leading strand, it can kind of synthesize the fragment and follow the replication fork. Nice and neat. Let's see here. So they call that the leading strand. So I'm writing, the leading strand builds a new DNA in the same direction as the replication fork. As replication fork. The lagging strand, you have to build it in the opposite way. So you have to build it this way, but the fork is unwinding the opposite way. So you always have to kind of go back and then build the gap you missed. So the lagging strand, for that reason, has to like make the new DNA here in fragments. And then those fragments are linked together with enzymes because it can only go this way for this strand down here. The lagging strand. The lagging strand must build the new DNA in fragments. I think they're called Lozaki fragments. There's a ligase enzyme that can kind of link the fragments together. Ligase can link the fragments. So that's DNA replication. I want to spend much more time talking about DNA making protein. That's why it's there. Now we need to replicate it for mitosis, but this is really the function of it. So to go from nucleotides to proteins, well, DNA will carry the instructions on how to make proteins. Pretty much you have one gene making one protein. That's usually the relationship. Genetic information in the DNA is conveyed in the linear sequences of the four bases, A, T, G, C. So the other idea we try to get across to students is that the gene and its polypeptide products are collinear. That's the other idea. The first big concept is the base pairing. Now we have this idea of gene and polypeptide being collinear. The gene, a collection of adjacent nucleotides that specify an amino acid sequence for the chain. The corresponding nucleotide and amino acid sequence are collinear. So I usually demonstrate collinear by giving an example. Let's say you wanted, instead of coding for amino acids, you want to code for letters of the alphabet or something. And you want to use numbers to code for letters. Use numbers to code for letters. And how many letters of the alphabet are there? There's 26 letters of the alphabet. And let's just use regular counting numbers to code for the letters. So you just need the first 26 numbers, 1 to 26, to code for the 26 letters. And let's say our code is the secret code that no one's ever used. Do you see the pattern? I think you do. Therefore, what is the code for Z? 26. Okay, and let's say this is my simple example. This is my code. Collinear is if I take my code, my numbers, and I'm coding for letters, 433. Okay, so that's BCC. Okay, and 211 is BAA, and 122 is ABB. So do you see how I did that? I went from left to right, and I decoded it because it was linear. I didn't have to skip around. I didn't have to go here, then here, then there. It wasn't random like that. That's what's collinear. Whatever your code is, it's in a row. They're in line with each other. So that makes it really easy for students to do. Okay. Now the problem we have is we're using the nucleotides. There are 4. There's 20 amino acids. So if it were a 1-to-1 relationship like this over here, one number codes for one letter. If it were like that for this situation, how many amino acids could you code for? Well, if there's only 4 nucleotides, you could only code for 4 amino acids. Now, do you see the problem with that? That's a very ineffective code. You're missing the other 16. So obviously, this being the code for that is not a 1-to-1 relationship. For example, A cannot be the code for, I'm making it up, histidine. It can't be because you'd be missing 16. And so what it is, it's not a 1-to-1 relationship, correspondence between nucleotides and amino acids. Basically, the genetic code, it's fully deciphered by 1966. Here's the code. It's not 1-to-1. We use triplet sequences for the nucleotides. Because when you have 4 nucleotides and you have triplet sequences, you have plenty of different permutations to code for the 20. In fact, you have over 60. You only need a 20. So for example, any of these 4 permutations, CUU, CUC, CUA, CUG, these are the mRNA nucleotides. They all could code for leucine. LEU, yeah. So you have more than you need. You needed 20 different codes, but when you put them in triplets, you have over 60. Okay, so this is the genetic code. So what we learn is use triplet patterns. Okay, and so what we see here is the genetic code is amino specifieds are coded are specified by triplet codons. All right, let's take a break. When we come back from break, we'll kind of learn how to use the code and decode it.
For the test, are you going to want us to memorize the codons?
I want you to use the table, but I'll give it to you, so you do not have to memorize it. So we'll take our 15-minute break, and see you in a bit.